In order to view specimens under the microscope, we need to prepare them. They're generally prepared by mounting a sample on a glass slide. How the slide is prepared really depends on the condition of the specimen, whether it's living or preserved, the aims of the examiner, whether we want to observe the overall structure or identify the microorganisms, or actually see whether they move or not, and also the type of microscopy available. We can't really prepare for scanning electron microscopy if we don't have a scanning electron microscope. So once we have a preparation, that mount, the prepared specimen, sits on the stage between the condenser and the objective lens. So right here. There are two general types of preparations: living preparations and then fixed and stained smears. So in living preparations, we can use a wet mount. That's where cells are suspended in a fluid and a drop or two of the culture is placed on the slide. Then we just drop a cover slip over the slide and place it on the stage for observation. When we use a cover slip, though, sometimes you can damage larger cells, or we might dry them out, or even contaminate the observer's fingers. So sometimes a hanging drop mount is a better solution. This is what we see below. There's a depression in the slide, and we suspend a sample from the cover slip and then use Vaseline to fix the cover slip in place. This allows a much more three-dimensional space in which we don't crush our specimen. Now, when we come to fixed or stained smears, there's a number of different techniques. Rather than dropping a liquid onto the slide or onto the back of a cover slip, we smear specimen onto a slide. The smear technique was developed by Robert Koch over 100 years ago, and we still use it today. The thin film is spread out across the slide. It's made from a liquid suspension of cells, and then we take that smear and air dry it. After it's air dried, we heat the dried smear by a process called heat fixation. This fixation both kills our specimen and attaches it to the slide. So these kinds of techniques can be used for longer-term study. Sometimes, instead of heat fixation, if we want to have our cells in a more natural state, we can use chemicals like alcohol or formalin to preserve the specimen. Now, these don't fix them to the slide, thus we don't alter the shape any. Now, most microbes are colorless, very small, and difficult to see. So we must develop them, much like photographs. When you first have the negative, although we don't really have those these days, you can't really see any picture on it. Once we develop them, we can illuminate what's on the film. So staining will increase the contrast and sometimes increase the size, so it makes the specimens easier to see. This is where we're going to apply colored chemicals to the specimens. The dyes can either become affixed to the cells through a chemical reaction, or they can be repelled by the cells. Dyes are classified as either basic, which are cationic and positively charged, or acidic, which are anionic or negatively charged. Because of these variances in charges, they can be attracted to different parts of the cell respectively. Negative repels negative, but attracts positive. Beneath these fixed or stained preparations, there are a variety of different staining types. First, let's look at positive versus negative staining. In positive staining, the dye is actually sticking to the specimen to give it color. So this would be the case of a negatively charged particle attracting the positively charged stain, and thus it becomes colored. Negative staining, on the other hand, is where the dye does not stick to the specimen. So negative repelling negative. Instead, the dye settles around the boundaries, creating a silhouette. We can see both these examples over here on the right. A positive staining where the specimen is colored by the blue dye, and a negative staining where the specimen remains colorless and is surrounded by stain. Nagrosin and India ink are commonly used as negative stains, and heat fixation is not required here, so we have less shrinkage and distortion of the cells. They maintain much more of a natural shape. As we'll see later, this technique can also be used to accentuate a capsule surrounding certain types of bacteria or yeasts. These techniques allow us to assess the cell's size, shape, and arrangement relatively quickly. So now let's look at simple versus differential stains. Simple stains require only a single dye. Examples include malachite green, crystal violet, basic fuchsia, and saffronin. Now, don't worry about what those are yet. You'll be introduced to them throughout the semester. All cells in a simple stain are going to appear relatively the same in color, but it can reveal information about shape, size, and arrangement. In this case, we're seeing positive, simple stains. Differential stains use two differently colored dyes. We might see purple versus red or blue versus pink. These two dyes are called a primary dye and then a counter stain. This allows us to distinguish between various cell types or various parts of a cell. Examples include a gram stain, acid-fast stain, and endospore stains. Again, don't worry about the details right now, just introducing the variety of stains available. Gram staining is a specific differential stain. It's the most universal and diagnostic staining technique for bacteria. Gram staining was developed over 100 years ago by a man named Hans Christian Gram. It allows us to differentiate microbes as either gram-positive, staining purple, or gram-negative, where they stain red. This technique can allow us to elucidate information about the cell wall structure of these microbes. We'll certainly cover this technique in our lab sections throughout the semester. Another specific type of differential staining is acid-fast staining. It's an important diagnostic stain. It differentiates acid-fast bacteria, pink, from non-acid-fast bacteria, or blue. It's been important in medical microbiology. For example, Mycobacterium tuberculosis, the microbe responsible for causing tuberculosis, has an impermeable outer wall that binds tightly to the dye carbofuchsin, even when it's washed in acid or alcohol. So if it stains when the acid is washed off, then it could be recognized as Mycobacterium tuberculosis. Now, some other different stains. The endospore stain is where dye is forced by heat into resistant bodies, the spores or the endospores. So it allows us to distinguish between the spores and the cells that they come from, or the vegetative cells, because they are producing spores. Endospore staining is very significant in medical microbiology, and we'll shed more light on that as we move through the semester. Now, there are some other specialized stains that are used to emphasize certain cell parts that aren't revealed by conventional staining methods. These examples include flagellar staining or capsule staining. We looked at capsule staining a little earlier, where we're using a negative stain to show if a specimen has a capsule. This can be useful in identifying microbes that do have capsules, as not all of them do. So it can help us classify them. In flagellar staining, we're adding a coating to the flagella so it can be resolvable, because flagella on their own are so tiny that we can't resolve them under the light microscope. So I know this has been a whirlwind through the various staining techniques. But remember, there are two basic types of preparations, the living preparations and then the fixed or stained smears. We can have fixed or stained smears that are either positive or negative staining. And then in the positive staining realm, we can have simple stains, differential stains, and some differential stains include gram staining and acid-fast staining. So that brings us to the end of the material for this chapter. Before we go, let's have one more concept check. What sort of microscopes do we commonly use in the laboratory? Simple, electron, phase contrast, or compound? You know from early on that simple is just one lens, and we don't use one lens. That would be like a magnifying glass, so we can cross that one off the list. An electron microscope, well, those are very expensive, and so we are probably not going to use them commonly in our lab. We can cross that off. Phase contrast and compound. Compound is the way to go. Compound are readily available, and you'll be using one for examination of specimens in our labs.